Hi! The objective of the session is to draw the structure of nephron in a simplified manner. Metabolic activities in animals release nitrogenous waste materials like urea, uric acid, creatine phosphate, ammonia, etc. If these nitrogenous waste material remain in the human body, it leads to toxicity. So we have excretory system to remove these waste materials. The most important organ of excretory system in human body are kidneys. Each kidney consists of millions of tubular filtering units called nephrons. The nephrons are surrounded by a tuft of blood capillaries and peritubular capillaries. It's generally difficult to draw the diagram of this nephron because it is just tubular structures and capillaries. Trust me, you just have to remember the letters U and Y. Just draw U's and Y's and let's complete the diagram. Let's draw 6 U's. A U here, 2 U's here, and a long hairpin like U here, and 2 more U's. Fine. Now let's link them together. So this part is clear for you. Now let us draw a double walled structure for Bowman's capsule. Clear the bottom part of it. Make this also tubular by drawing lines. Now this end will meet a stem. So this is a stem, which is actually the collecting duct. Link these tubes here. This is the basic structure of a nephron. So this part is Bowman's capsule. This coiled region is proximal convoluted tubule. Hairpin like region here, it is loop of Henle, and the next coiled structure is distal convoluted tubule which joins to the collecting duct. Now this collecting duct will be having the branches of many nephrons connected to it, so we can draw few branches arising from the collecting duct like this. Basic structure. Now we have to draw the blood vessels. Let's start with the blood vessels. This is a branch of renal artery and it is called as Efferent Arteriole. Efferent, A for apple. So this is Efferent Arteriole and this enters into this Bowman's capsule. It forms a tuft of blood capillaries here, and this tuft of blood capillary later comes out as efferent arteriole. Now when you draw that, there should be some difference between efferent and efferent arteriole. This efferent arteriole, the lumen of it is slightly bigger compared to that of efferent arteriole. So let's draw it like this. Efferent arteriole, the lumen is smaller compared to that of efferent arteriole. Now what happens here? It forms a tuft of blood capillaries. So just fill the space with another U. And how will you draw the blood capillaries? Just draw few splits over here. I'll just expand it this side so it will be clear for you. So what you'll do? This is efferent arteriole. This is efferent arteriole where the lumen is much smaller, and this is the glomerulus part, and just have to show some splits here which gives you the effect of blood capillaries. Okay, so this is how the glomerular part need to be drawn. So this area is the glomerulus. Now what happens to this efferent arteriole? They form branches. They form branches and completely surround this entire tubular part of the nephron. So I'll just draw it like this. Draw a line, and one of this line has to go parallel to the loop of Henle and then just comes out like this. Okay, so I have drawn a line from efferent arteriole. It has come around this loop of Henle, parallel to loop of Henle, and then it has come out. Now see, all these blood vessels we have to draw double lines. In biology, always draw double lines for the blood vessels. So we draw now. You have to draw few Y's over here, which shows the peritubular capillaries. So just show or draw Y's. So this Y will connect the efferent arteriole with this parallel blood vessel. Okay, now let us draw double lines here. Just clear this part and draw a line. Same thing here. Just make double line. Done at this part. Let's come here. Same thing. Just draw Y, a Y, and the Y can be slightly changed in this manner. So for everything you have to draw double lines because they are representing blood vessels. We can shade these blood vessels by using a pencil. Clear? Now labeling and also writing the title below the diagram. Labeling starts. This is collecting duct. Highly coiled structure here, which joins directly to collecting duct, is a distal convoluted tubule. The blood vessel that runs parallel to loop of Henle is vasa recta. The double-walled structure where filtration happens is called as Bowman's capsule. This is a branch of renal artery. Renal artery forms the tuft of blood capillaries inside Bowman's capsule, which is called as glomerulus. Highly coiled region immediately after Bowman's capsule, it's called as proximal convoluted tubule. and hairpin length part of the tubule of nephron, it's called as loop of Henle. So this is how you can draw structure of nephron in a very simplified manner, and this is for grade 10. As per grade 11 syllabus, few more additional parts need to be labeled. Let's do that. This can be marked as Efferent Arteriole, this is Efferent Arteriole, these are the peritubular capillaries, this is the descending limb of loop of Henle, and this one is the ascending limb. Hope the diagram is clear. The tips and tricks for drawing a biology diagram I have uploaded in my previous video, so you please watch that. You'll get a clarity on that. Thank you.